Hi, my name is Wei, Associate Director of Code Research in n-Lorem. My major work responsibility is to lead the team research to develop allele-selective ASO to treat nano-rare patients. Today, I will be talking about MAPK8IP3. We also call it as JIP3. The full name is very long, Mutation-activated protein kinase 8 interacting protein 3, which is one of our patient case for developing ASO therapy. JIP3 is an adapter protein of the kinase 1 complex, and it plays an important role in the axonal transport of cargo. Mutations in this gene have been linked to the severe neuronal developmental disorders. This including developmental delay, intelligence disable, ataxia, and seizure, and even visual impairment. In our study, we examined a patient who carried a missense mutation in MAPK8IP3, especially chromosome 1714C2T, which causes an arginine to cysteine substitution at position 578. We also call it as R578C mutation. This patient presented with the dystonia, growth, motor delay, and overall developmental delay. What we found is that this mutation behaves as a toxic gain-of-function mutation. It alters the intactome of the JIP3, disrupts exon transport of the later exosomals, and increases signaling through the Zhang, or we also call it Zhang signaling. As a result, it triggers apoptosis. Interesting, it also disrupts dopamine receptor 1 signaling, but does not affect dopamine receptor 2 signaling. Finally, we develop ASO to reduce of JIP3 to offer potential therapeutic strategy to treat MAPK8IP3 mutation-induced rare disease. So first, let's know what is the MAPK8IP3. This gene encodes JIP3, a scaffold protein essential for exon transportation, lysosomal trafficking, and Zhang signaling. Variants in the MAPK8IP3 cause a spectrum of the neuronal developmental disorders. The very important step is to determine loss of function or gain of function before we can use ASO therapy. We found that this R578C help activate Zhang signaling, mislocalize the JIP3, impair the late endosomal trafficking, and disrupt dopamine receptor D1 signaling, leading to apoptosis. Based on this evidence, we established R578C MAPK8IP3 mutation as a severe toxic gain-of-function mutation. Importantly, reduced JIP3 levels with a non-allele-selective ASO rescues all pathogenic phenotype. These findings define the mechanisms of the R578C toxicity and highlight RNA-targeted strategies as a promising therapy for MAPK8IP3-related disorders. Okay, next, let's look at the results to support our findings. First, we used the HeLa cells as a research model. Endogenous JIP3 was nearly undetectable, so we can transiently transfer the wild-type and mutation JIP3 in the HeLa cells. Because JIP4 can compensate, so we also reduce its expression using the siRNAs. So you can see here the data of expression of the both wild-type and mutation JIP3 induced apoptosis, which represented by the Cas3 and 7 activities. However, when JIP4 was reduced, wild-type JIP3 no longer triggered cell death, while mutation JIP3 continued to drive apoptosis and activate Zhang signaling. Also, the co-IP data showed that JIP3 interacts with the JIP4, likely forming heterodimers, but the mutation JIP3 co-precipitate less with JIP4. This stress mutation JIP3 acts independently to cause cytotoxicity. So this result you can find in the Western blot, the protein measurement. We also found mutation JIP3 can disrupt the endosomal trafficking. So you can see here, site 3 labeled ASOs moved slowly and traveled short distance, failed to accumulate properly at the peripheral nuclear region. This result demonstrates that the mutation JIP3 impairs endosomal mobility along the microtubules, also contributing to the cytotoxicity. To validate these findings in a patient-relevant system, we used fibroblasts from a patient carrying the R578C mutation. These cells express mutation JIP3 and show reduced proliferation and increased apoptosis compared to the control cells. Unlike control cells, patient fibroblasts did not respond to LPS stimulation, and the phospho-Zhang was not increased by the LPS treatment, but we could still see a higher Zhang signaling activation in the patient cells compared to the control cells. Further, we also observed that endosomal trafficking was impaired in the patient cells. Compared to the control cells, site 3 labeled transferrin failed to accumulate in the mTOR. This is the microtubule organizing center in the patient cells compared to control cells. Next, we also checked the endogenous cargo FYP labeled TLR4, which is known for endosomal trafficking cargo. However, TLR4 failed to co-localize with RAB7, while with the RAB5 remain intact in the patient cells. It indicates that JIP3 delayed late endosomal mobility. Together, this data show that the mutation JIP3 reduces cell proliferation, enhances Zhang signaling, and disrupts endosomal mobility in the patient fibroblasts, confirming cytotoxicity through the multiple mechanisms. Mutation JIP3 causes neuronal disease. Therefore, we checked the mutation JIP3 functions on patient iPSC-driven neurons. In patient neurons, JIP3 well expressed, while JIP4 remained low. This neuron showed enhanced Zhang signaling and reduced cell viability compared to the control neurons. Next, we imagined the neuron super-resolution microscope imaging showed impaired axon trafficking. We found fewer PS ASO localized with RAB7 in the patient neurons compared to the control neurons. It indicates late endosomal mobility defection in the patient neurons. Those results demonstrate that the mutation JIP3 disrupts axon trafficking, enhances Zhang signaling, and reduces neuron survival, confirming R578C mutation as a severe toxic gain-of-function mutation in the neurons. There was a physician reported to use L-DOPA can relieve MAPK ATP3 mutation-induced disease symptoms. As we all know, L-DOPA is related to the Parkinson's disease and dopamine receptors signaling. So interesting, we found both healthy and patient fibroblasts expressed dopamine receptors. Using selective agonist for D1 SKF and D2 RAP, we found D1 stimulation increased cAMP in the control fibroblasts, but failed to do so in the patient fibroblasts. D2 signaling remaining dose-dependent and intact in both cell types. We also see a higher basal level in patient cells than control cells, and through blocking A beta-1, beta-2 AR receptors normalize the increased basal cAMP in patient cells, confirming that AR receptors drive this effect. D1 agonist SCH only worked in the control cells, but not work in the patient cells, highlighting a selective D1 signaling disruption caused by mutation JIP3. Those results indicate a specific defect in the D1 receptor function in patient fibroblasts, providing insight into JIP3-induced abnormal dopamine signaling. Next, we look further in the iPSC-driven neurons. In patient iPSC-driven neurons, both D1 and D2 receptors were highly expressed, but the CMP level was not induced by D1 agonist SKF, regardless of the D2 activation. Interesting, when using the beta-1, beta-2 AR antagonist blocked the AR receptor effects, we only saw D1 agonist SKF worked in the control neurons, but not in the patient neurons. It confirmed that D1 signaling remained defective in the patient iPSC-driven neurons. Co-IP experiments showed that D1 interacts directly with JIP3, and this interaction was increased with the mutation JIP3. This suggests that mutation JIP3 over-engages D1, interfering with normal signaling. Those findings demonstrate that mutation JIP3 selectively disrupts D1 receptor signaling in patient neurons, providing mechanistic link between the JIP3 mutation and the dopamine pathway dysfunction. Next, we want to know if we can treat this mutation. To assess therapeutic potential, we developed the non-allele-selective PS-more gapmer ASO targeting both alleles of the MAPK8IP3. This ASO recruited RNA SEH1 to degrade the targeted mRNA, effectively reducing expression of JIP3. In patient fibroblasts, the ASO reduced the mutation JIP3 expression and restored proliferation in a dose-dependent manner in the patient cells. Of course, non-allele-selective ASO also reduced wild-type around 60%, but this was tolerated by cells. Then we also checked the protein level. We found ASO reduced Zhang signaling. You can see phosphor Zhang activation is reduced. Further, endosomal trafficking was also a normalized ASO in patient cells with the ASO treatment, Cy3 transferrin accumulation in the late endosome, and co-localized with RAB7, similar to the control cells. Those results demonstrate that non-allele-selective ASOs can effectively suppress mutation JIP3, restore proliferation, and normalizing signaling, and rescue endosomal function. Next, we further look at the ASO effects on the neuron. We applied the 3 lead ASOs to patient iPSC-driven neurons. All of 3 restore cell proliferation to control level. ALS2 is most effective reducing mutation JIP3 mRNA by over 80%, while wild-type JIP3 was also reduced. This was tolerated, and the patient cell viability increased significantly. Dose-dependent treatment with ALS2 decreased the JIP3 and attenuated Zhang signaling activation. Also, hazard ratio analysis showed that mutation JIP3 increased the neuron risk, which was reduced by AS2. Importantly, D1 receptor coupling was restored in response to the SKF stimulation. These findings confirm that non-allele-selective ASO rescue patient neurons from cytotoxicity, restore the signaling, and support their potential as a therapy strategy for the MAPK8IP3 mutation-related disorders. In conclusion, this study reveals for the first time that the mutation JIP3 R578C causes abnormal enhancement of the Zhang signaling pathway, deficient cargo endosomal movement transportation, and a disruption of dopamine receptor response. This abnormal observation eventually led to cell death, demonstrating that the mutation requires a severe toxic gain of function. It also further expands how this mutation leads to the neurodegenerative disease. Finally, this study also provides a reliable and effective ASO treatment method, which can significantly inform a potential clinical treatment. Finally, I want to give great thanks to the Wolverine Foundation to support this project, And also thanks to Joshua Nico from Illinois to give the consultant on this project. Also give the great thanks to the Columbia Stem Cell Core people to provide the patient-related cells. Thank you so much for your listening.